All right, so thank you very much for your patience. Thank you very much for inviting me, Melissa and everybody else. I'm really excited today to talk to you a little bit about what I believe to be let's say, the good news and the bad news stemming from recent progress that we have seen in genomics and genomics research. So when I started working in this area, it was very interesting to me how costs of whole genome sequencing were dropping, how fast they were dropping, so much faster than what Moore's law will predict. I'm sure many of you have seen this graph before, but it is clear to me that we really went from what it sounded like a futuristic venture, like the Human Genome Project, which took 13 years, $3 billion in investment to sequence one human genome, to an increasingly affordable technology, right? So we have companies in this space like Illumina or Oxford Nanopore that can fully sequence a human genome in a matter of hours for costs in the order of $1,000, if not less. And so this is enabling a number of initiatives aimed at sequencing large amounts of individuals worldwide. So in the UK, we have a project to sequence 100,000 genomes in the US. Of course, everything has to be bigger. So we have a project to sequence 1 million people. And in general, we have seen an increasing number of individuals having access to genetic testing, getting their genome sequenced. Right? So why is whole genome sequencing such a big deal, right? So for decades, we have been doing genetic testing, and we have used what typically is referred to as genotyping, which essentially are in vitro processes, in vitro tests for essentially looking for known genetic differences using a set of markers. So you go and look for the presence of, let's say, a genetic mutation, for instance, right? But now, the ability to completely sequence a human genome gives hope to scientists so that they will be able to study essentially the entire genome sequence and understand what they don't know and how the genome as a whole works. So how the different genes work together, let's say, to direct the growth, the development, the maintenance of an organism, right? So an important point that I want to make is that the whole genome sequencing is already helping researchers to gain a better understanding of diseases and their relationship with genetic features. And this is already bearing fruits in, let's say, the clinical setting, not only in the research setting, which maybe you have heard of a little bit more, like cancer, research on cancer or Alzheimer's and so on. So there is more and more news coming from our hospitals where sequencing has saved lives, right? So we have been able to diagnose and cure patients thanks to whole genome sequencing. However, this kind of genomics revolution, if you let me use this expression, is not only happening within the walls of our research labs, our hospitals. It's something that is entering our society, let's say, right? So, and I chose to pick the example of Angelina Jolie, who was diagnosed with genetic predisposition to breast and ovarian cancer, and her decision to undergo double mastectomy and hysterectomy really put, let's say, genetic testing in the spotlight, if not for the debate that it generated. There's also an increased market of companies that are commonly known as direct-to-consumer genetic testing companies, like 23andMe is definitely the most known one. So, I mean, they essentially sell products marketed at about $100 or so, where there is no involvement of a doctor or health professional. So customers essentially get a DNA sample, DNA kit. They deliver a saliva sample. They mail that in, and after a few days, they have access to a website that displays the users the results of this genetic testing. So they can learn things like whether or not they turn red if they drink alcohol, or whether or not they carry some traits, so some genetic variants that are known to cause some genetic disease, or the predisposition to certain diseases, or how they would respond to certain drugs, for instance, like warfarin, a common blood thinning drug. So in general, as I said, there is an increased number of individuals that do get access to genetic testing, motivated by medical reasons or personal curiosity. So, for instance, they want to find out about their genetic ancestry, so their ethnicity, and they learn that they are 9% Finnish or something like that. So there are companies like, again, 23andMe or Ancestry.com that offer these services. And again, like 23andMe and others that also allow you to find what they believe to be your relatives in genetic terms, let's say. So for instance, like your third cousin or your fifth cousin, looking at other customers of 23andMe. And keep in mind that 23andMe has more than 1 million customers. So it's a non-negligible size, right? So now having said that, I am a computer scientist, right? So I'm not a geneticist. I don't work in biology. And to me, whole genome sequencing really sort of means turning this, like a double-stranded polymer of nucleotides, into this, right? Data, right? So this is the output of, I think, the Lumina sequencing machine is a SAM files. And it contains reads, like these nucleotides, these letters, and some additional information. But with any kind of data, we computer scientists and bioinformaticians, we need to store data somewhere. We need to essentially provide tools for researchers or for clinicians to search this data, perform queries, computation, and so on, right? But it's clear now, as a privacy researchers, it's clear that not all data are created equal. So some are maybe a little bit more sensitive than others. So I'd like to maybe discuss a little bit what I believe to be the specific features of genomic information. So it is clear, I would say, that genomic data is a treasure trove of sensitive information, in that if you have access to my, let's say, sequence genome, you can quickly figure out my ethnic heritage, maybe my predisposition to diseases, maybe even including mental conditions, and so on, right? It is also, the genome, let's say, by design, is a unique identifier, right? It's the ultimate identifier, and as such, also considering its hereditary nature, it becomes extremely hard to effectively anonymize or de-identify genomic information. And this also means that if you leaking one's genome essentially implies leaking the genome of close relatives, right? Which also means that the sensitivity of this data is almost perpetual, right? So even, let's say, 50 years from now, if you get a hold of my genomic information, I might not be around, or I might not be able to complain, but my kids and grandkids might, right? So because the genomes are so close to each other, right? So in a sense, it's kind of, it is a unique threat here, in the sense that with other kind of sensitive information, like, I don't know, if someone compromises your credit card information, your password, or even your social security number, you have mechanisms to recover from that, right? And you can even issue new social security numbers, I guess, but you cannot really issue a new genome to yourself, right? And as I said, its sensitivity extends probably beyond one person's lifetime. Okay? So this is sort of creating maybe a conflict between privacy and the greater good. The fact that research in genomics is extremely great news for us, is helping us fight cancer and so on. And maybe I'm stating the obvious here, but advances in genomic research is highly dependent on the availability of data, on data sharing. Right? It's an important asset for researchers worldwide. In fact, in certain countries, access to public funding for genomic research means that you have to make a best effort to make data available to other researchers. So if you sequence thousands of individuals, you should make this data available. Right? And even if costs drop quite quickly, it's unfeasible to assume that every research lab has the ability to sequence thousands of people. Right? So, and let's not forget that a lot of the studies, like what we call genome-wide association studies, where really you try to understand if a variant, a genetic mutation, let's say, is associated with a trait, really require access to large number of genomes. Okay, so this is sort of motivating a new research community, which I'm going to call the genome privacy research community, which essentially has been doing in the past few years two things. One, studying the privacy issues, carefully analyzing those issues, and two, designing privacy-preserving genetic testing algorithms. And there is this amazing website, genomeprivacy.org, that has been keeping track of essentially all papers published in this area. So if you want to know a little bit more, this is a great resource. So in the rest of the talk, I'm just going to give an overview of what I believe are the main results in this area, starting with studying privacy issues. And these are, I'm going to select, let's say, the most visible papers, maybe the first one. So if you're in the audience and I don't cite your paper, I apologize. I'm going to blame time constraints. Okay, so the first one I want to talk about is relates to de-anonymization of genetic information. So there was this kind of high-profile paper back in 2013 published by Science, where Melissa Gimreck et al. Showed essentially the feasibility of re-identifying DNA donors from a public research database using information publicly available on popular genealogy websites like Ancestry.com and other available information. So you have some part of data that is supposedly anonymous, like this research database Personal Genome Project, and you have some other information that contains identifiers. In this case, was actually down to family names, so like last name. And now you correlate this data using the fact that genetic information is hereditary by nature. And so they were able to pin down last name of anonymous donors in 40% of cases. So for those of you who are familiar, let's say, with the famous Netflix de-anonymization paper, I mean, the techniques are sort of similar. Okay, so de-anonymization is hard. We should, if we go that route, we should be extremely careful. But how about aggregation, right? So aggregation is also very hard in genomics. So, I mean, there's been a couple of high-profile studies that show that, even statistics from allele frequencies, so just, let's say, reporting how many donors exhibit certain characteristics can be used to re-identify genetic trial participants. So for instance, test for the presence of a specific individual, let's say a victim, in a group. So even aggregating data, so doing privacy by aggregation, it's something that should be done extremely carefully. And I mean, these are just two examples of the attacks that have been presented in this space. Another study that I want to cite is one that comes from EPFL that looked at the concept of kin privacy. As I mentioned before, if your genome is leaked, then privacy of your close relative is also hurt. And so they were motivated by this kind of real-life case of Henrietta Lacks, who was a cancer patient that died many years ago. And without her consent, researchers worldwide have been using her genome for cancer research. Okay, so again, she's no longer with us, but her family still is, right? Her descendants, right? So this paper tried to really quantify how much privacy relatives lose when one's genome is leaked. And if you're interested a little bit more in understanding, besides this genomeprivacy.org website, there is a couple of nice surveys, including ours. Sorry for for the shameless self-promotion, that kind of look at all the results in this space. I also want to make the point that this studying privacy issues is not something that has been done only by computer scientists, looking at these kind of characteristics of data, but also social science and anthropology and psychology and ethics communities have looked, for instance, at studying individuals' fears and concerns related to genetic testing and genomics. Okay, so as I mentioned before, these kind of fears may be related to disclosing of sensitive information. So they usually relate to fears of being discriminated, let's say in the workplace or by health insurance providers. So researchers have looked also at perceptions of people, as well as reactions to learning test outcomes, like having predisposition to get cancer or Alzheimer's, and also learning results that you did not really expect, like this case where someone learned they had a half-brother, and this ultimately led to their parents getting divorced. Okay, so now going to the second line of work. So how can we enable genetic testing while preserving privacy of individuals? Okay, so one important result in this space came back in 2013 when Aaron Johnson and Vitaly Shmatikov investigated the feasibility of applying differential privacy for genome-wide association studies. So differential privacy, for those of you who are not familiar with it, consists in adding noise to a data set with the goal of supporting statistical queries. While you can provably preserve the privacy of the individuals whose information is contained in the data set. Okay? So in this case, you trade off accuracy with privacy, and you can support queries like computing the number or location of SNPs, so these genetic variants associated to a disease or significance, correlation. And the results, they do suffer from an accuracy loss, but they showed us that this was a viable avenue. There's also been a lot of work, and I think we will hear about it from the next two speakers, on computation over encrypted genomes. So there's been a lot of work coming out of Microsoft, as well as people in Estonia that have been using what we, in cryptography, we refer to as multi-party computation, using secret sharing. So essentially, you have multiple parties that, and you assume that the majority of them doesn't collude with each other, and then you can, distribute this computation in such a way that, the input is not disclosed, but only the output. Another line of work deals with personal genomic tests, so more similar to, let's say, the 23andMe scenario, where, you have individuals who want to have access to their genome, and they want to have access to genetic testing. So they want to allow doctors, clinicians, testing facilities to do these tests, right? So in this setting, privacy for us means that individuals should retain control of their sequenced genome, but still allow doctors, for instance, to run genetic tests in such a way that the genome itself is not disclosed. So they don't have to forego their privacy, let's say. Only the test output is. And I'm going to give a little bit more details here. But it's also important to recognize that in some cases, also the test specifics should be kept confidential. So if you have a pharmaceutical company, let's say, that designs a certain genetic test to, for instance, assess the right dosage for warfarin, then this test specific is kind of like their secret sauce. So it is intellectual property, and it should not, this should not be revealed. So there is two main approaches in this space. One essentially relies on cloud storage providers that are semi-trusted in the sense that they're not assumed to collude with the testing facility itself. So the idea is that we have genomic information, it gets encrypted and stored onto this cloud, this storage and processing unit in this picture. And now a testing facility can access, can perform, for instance, disease risk computation without learning the actual data, but just the test output. And these are maybe the first papers in this space were published back in 2013, but there is much more. And the second kind of approach is to actually let users keep their sequence genome. So not storing it onto the cloud, but for instance, giving it to the users on a dedicated device, for instance. Okay. So now what we do actually is to reduce genetic testing to evaluating a function, right? Which we can do, right? So you want to test for presence of certain mutations. You want to test, so you want to find some patterns. These you can express as a function, right? And in cryptography, we know how to securely evaluate a function in that only the test result is disclosed. So only the output of the function is disclosed. So nothing is revealed beyond the test result. And so here there is a number of cryptographic techniques that we can use, like garbled circuits, homomorphic encryption, private set operations, and so on. So a lot of papers have been using these techniques to support genetic testing, like testing for SNPs, doing this personalized medicine style tests, or paternity ancestry testing, and so on. And in a sense, this kind of started with our CCS paper in 2011, where we showed how to essentially scale up to whole genome sequencing. Okay, so now I'm kind of getting ready to wrap up and leave the floor to the other panelists. So I also want to point out that there is still a lot of open problems in this space. And essentially, one of the big things that we have to discuss is that maybe encryption by itself or cryptography by itself in this space might not solve all the problems, right? And the clear one is the fact that encryption, we cannot guarantee the security of any encryption scheme, essentially, that are used today past 30 or 50 years, right? So even if I use AES to 156-bit, this is assumed to be secure for 30 years. And again, the sensitivity of genomic information will not degrade in 30 years, right? So we need to think carefully about this. And of course, other problems relate to efficiency, and reliability, availability, which are challenged by the use of cryptography, right? Not to mention that cryptography itself makes it significantly harder to deal with sequencing errors, which do happen today. So you have, when you read the genome, you have insertion, you have deletion, you have actually sequencing errors. So instead of reading an A, you read a G, right? So designing algorithms that are resilient to errors is much harder when you're operating over encrypted data, okay? And of course, there's also the consideration that when you do involve users, how much understanding you require from them in terms of protecting this data, saving keys, saving passwords, explaining to them what it means to provably protect their privacy, and so on. All right, so this concludes my talk. I also want to thank my collaborators and my funders, and I'll be happy to answer your questions after in the Q&A session. All right, thank you.
So thanks, Emiliano. That was a great talk. Our next speaker is going to be Xiaoqing Jiang, who's an assistant professor in the biomedical informatics department at UCSD. He's also an associate editor of the BMC Medical Informatics and Decision Making, and on the editorial board of the Journal of American Medical Informatics Association. And today he's going to tell us about some of the workshops he's been running on looking at genomic privacy, among other things.
Thanks, Melissa, for introducing. So I'm going to talk about iDASH privacy and security workshops, a community effort to access the state-of-the-art privacy and security models in genomic research. So as Emiliano has just mentioned, the DNA data is important to genomic research, biomedical study, and it's even becoming part of the electronic health record. But at the same time, it also highly sensitive. It brings a lot of issues to privacy and security. So Lin and Altman in 2004 showed only 75 SNP is enough to re-identify a unique individual. And there are many other examples, for example, like the surname identification by Genome paper, and recently another paper shows that even 3D face can be reconstructed from the DNA data. So the grand challenge here is really how can we analyze genomic data in a way that preserve the privacy of the data donor or the owner without undermining the utility of the data or impeding its convenient dissemination. So there's really a trade-off between privacy and utility. We can safeguard the computation process using secure primitives, but it has computation costs. We can add noise to the outcome to mask sensitive information, but introduce artifacts may render the output into useless information. And the question here is, can the state-of-art technique to be used to support biomedical research in practice? So iDASH is one of the national centers for biomedical computing centered at the University of California at San Diego. We're the only center that focusing on integrating data for analysis, anonymization, and sharing. So we've been hosting this workshop several years in a row, and recently we have been turning this workshop into a competition. And we try to use real study to understand the impact of data anonymization and the latest secure models to real-world biomedical studies. We try to use real human genome data and high dimension at a practical scale to evaluate the solutions. We seek solutions that balance and trade off the privacy and the utility. So in 2014, we hosted our first workshop on UCSD campus. And the first competition is to evaluate how effective the best privacy protecting technology can protect human genomic data and analysis result. In this workshop, we focus on the challenge to protect sharing aggregated SNP data, allele frequency in specific, and the top K SNP identification as query result for GWAS study. So we have a number of teams participating and quite a few online attendees. And we have two challenges. The first challenge is to understand the privacy utility balance achievability when public released SNP data are anonymized for a realistic GWAS study. So we define our utility as the chi-square association test in terms of significant SNP before and after anonymization. And we used one of the strongest re-identification statistical attack, the likelihood ratio test, to access the resident of the attack model after anonymization. So the attack model looks like this. So given allele frequency of individual of interest X at each position J, we compare it with the allele frequency P and P hat of the case and the control group. When accumulate, we can tell whether an individual is more likely to be in a case group. If there's a highly likely that the individual is in a case group, then the identity is revealed. So we implement the online tool for the participant to access their model. And the utility is evaluated on the case control association chi-square test. So at each SNP, we have a minor and a major allele, and given case and the control group, we have a 2x2 contingency table, and we can derive the expected contingency table accordingly, and then the difference between those give us whether the SNP is significant. The second challenge is not to anonymize data, but instead to protect the query result. So our goal is, given a privacy protection standard, evaluate how much utility, name how much top K significant SNP in terms of the plain text operation can be preserved after the anonymization is inserted to the outcome. And our utility is the percentage of the top K most significant SNP in terms of chi-square test. So K ranges from 1, 5 to 10, and so on. And our privacy criteria is differential privacy, and we set a privacy budget equals to 1. So differential privacy is a privacy criteria proposed by Dr. Cynthia Dwork at Microsoft to quantify the information leakage of statistical information release. And we prepared the data from 2 public domains. One is from the Personal Genome Project, and the other is from the HapMap project. And for each of the challenge, we prepared 2 data sets. One is released for the participant to tune their algorithm, and we reserve the larger data set for evaluation. For the first computation, we check the utility based on true positive rate, false positive rate at different cutoffs for the chi-square statistic. And then we also check the vulnerability of the anonymized model based on the re-identification power at 0.95 significance level. Overall, the McGill University demonstrated most balanced solution. For the second computation, we are checking on average of 1,000 iterations, how many top K SNPs are returned of the anonymized algorithm. Because differential privacy has some stochastic property, so this number is a mean of 1,000 run. As you can see, UT Austin team lead the computation. And after computation, we concluded that it still remains a challenge to do privacy-preserving data sharing of aggregated human genomic data while maintaining the utility of GWAS study. Even for a loci of a few hundred SNPs, the utility is largely damaged after we enforce the privacy protection. It is unlikely the current technology will scale well for the whole genome. But on the other hand, the privacy technology shows good promise on publishing outcome like GWAS-like analysis. High accuracy has been demonstrated only when only a small number of significant SNP are queried. And this is well aligned with the data computation model to release only the data to the user. So motivated by our success of the first year, we hosted a second workshop, and this year, in 2015, we switched the gear to secure genome analysis computation and to foster research to address secure outsourcing and secure multi-party collaboration in biomedical studies. We put 2 main application scenarios. One is for secure genome-wide association study. The other is for secure genome computation based on comparison based on Hamming and added distance. We have expanded number of teams participate, and the event was reported by Nature News and Genome Web. 11 teams from 12 institutions participate in the competition. For the first competition, we are seeking solutions of homomorphic encryption-based protocol to analyze encrypted DNA data on untrusted server. And the first task is to calculate minor allele frequency and chi-square statistic. Because minor allele frequency requires comparison and chi-square statistic requires division. These are not trivial operations in homomorphic encryption. And the second task is to calculate Hamming distance and added distance. And the protocol should return only the encrypted result. For example, the minor allele frequency, chi-square statistic, and distance, which only can be decrypted by the owner of the data. And the second task is to run solutions to enable 2 parties to work together to perform a genomic analysis across their DNA data set without actually exposing their data. The first task is to develop a distributed cryptographic protocol to securely aggregate minor allele frequency in 2 data sets and calculate chi-square statistic of the given SNPs. And the second task is to develop a distributed protocol to securely compute Hamming and added distance between 2 sequences in remote institutions. So for each of the submission, we required participant team to submit their solutions in preset virtual machines, either on a single server for the first task of into 2 servers located at UCSD and Indiana, respectively. And we recorded the memory usage, runtime, and communication of these solutions. So for the first task of the first track in the homomorphic encryption, Stanford and MIT team demonstrated the most efficient solution in terms of minor allele frequency and chi-square statistics. For the Hamming distance calculation, there are 3 teams participating. All of them shows accurate result comparing to the plain text operation, and the IBM team demonstrated the most efficient solution. For the approximate added distance, Microsoft team demonstrated the most accurate result and lead the competition. We also evaluate the SMC solutions in a similar way. For the chi-square statistic, we evaluate them on a large data set, and solutions proposed by University of Notre Dame and University of Maryland are very close to each other, but Maryland team does not utilize a third party. And for the Hamming distance calculation, University of Virginia, Maryland, UCI all proposed pretty good solution, and Virginia has a balanced performance in terms of communication, memory usage, and time. For the last track in the added distance, we evaluate the solutions over 100K variation sites, and UCI has an overall balanced solution. So we're very excited, and it's a great pleasure to award the winners with a big check. Thanks for Human Longevity for the generous support. And after the workshop, we think we are moving much closer to practical usage in a few aspects. For example, in analyzing encrypted DNA, Hamming and added distance approximation can be done within 10 minutes over 100K sequences. And secure collaboration over the internet for the GWAS study can be done in a few minutes for a few hundred SNPs. And for the Hamming and added distance, within 20 seconds on a 10 megabyte bandwidth, we can finish the calculation between Indiana and San Diego. So we're really close to protecting some of the genomic analysis tasks. However, there are still a lot of gaps needs to be closed. Full-fledged GWAS still not efficiently done on encrypted DNA, and due to the challenge of performing divisions and comparison and sorting. And HME, homomorphic encryption, requires multi-gigabyte of memory in our computation. And we also see SMC solutions transmitted multi-gigabyte of data across internet for analyzing even just 100K length of sequence. Operations that usually can be done within seconds takes minutes, even hours. And accurate added distance is still way off the table. So motivated by the previous two workshops, we are hosting a third workshop this year. Actually, we are changing our location from San Diego to Chicago this year in November to co-locate our event to one of the biggest medical conference, biomedical conference called AMIA. And so we try to engage more human genetic researchers to join our community and provide more use cases. And so far, we have already engaged more than 50 teams sign up for the competition over 13 countries. And we're receiving a lot of questions on weekly basis. And we are very excited to obtain the solutions on August 31st and evaluate them. And this year, we are trying to combine the themes from the previous 2 years. We set up 3 tracks. The first track is similar to the competition in the first year. We try to solicit for solutions to protect genome query service to protect its outcome. So we're trying to use Beacon as example. Beacon is a genome query service that answers whether a database contains a certain variation in its database. Recently, Stanford faculty Dr. Bosmanti has demonstrated just using this query, we can identify individuals in the presence of this database. So we try to develop solutions to mitigate that risk. And the second and the third track is inherited from the last year, but we are trying to expand it. For the second track, we are going to solicit solutions to search similar cancer patients across organizations. So this time we require much elevated added distance calculation because the previous approximation might not work in our set scenario. And the last task is to test the genetic diseases in encrypted data. And we're trying to expand this to the full-fledged genome of 5 million. And lastly, I want to thank all my co-organizers, Dr. Xiaofeng Wang, Haixu Tang, Shuang Wang, and Lucila Machado for their support. And the previous 2 years, we have been lining up with BMC Medical Informatics and Decision Making to publish a special journal. And this year, we are lucky to have a medical genomics lined up to publish this special issue. Thank you for listening.
Interesting talk. So our last speaker is It's going to be Kim Lane, who's actually a postdoc here in the cryptography group in Microsoft Research. So Kim joined us from Berkeley in the math department, but in the last year he's been becoming our expert on fully homomorphic encryption, and in particular building the library that was, I think, recently released. But so he'll tell you a lot more about that.
Does this work? Okay, works. Can you hear me? All right, great. So thank you, Melissa. So the title of my part of this talk is Private Predictions with Homomorphic Encryption. So in the previous talks, we heard about some of the problems associated with well, for example, genomic data, but there are often similar problems with other types of medical data also, and I want to propose here or demonstrate some possible solutions to some possible concrete solutions to solving such problems using, in this case, homomorphic encryption. Like, our group is at the moment very strongly focusing on computation on encrypted data techniques, different ways of doing applying functions to somehow private data in such a way that no extra information is leaked here. And one of the techniques that we've been using is homomorphic encryption. So here I'm going to show some results about that. So the sort of first part of this talk is explaining what we mean by privacy in prediction. It's maybe already sort of obvious from these previous talks, but one of the ways of how we like to think about this is that since Microsoft likes to provide services to other companies, we're thinking of like a prediction service that we could set up, which would do maybe medical prediction, genomic prediction, some maybe financial prediction. And we would somehow need to convince people, we would somehow need to convince other organizations, other companies, medical centers, hospitals to actually send us data to be processed. If we developed some models, for example, for like medical or genomic or financial applications, I mean, we need to somehow be able to convince our customers, potential customers, that they can indeed trust us with their data. And you just learned that this is possibly a very difficult task, especially in the case of the genomic information, because in that case you have also these issues related to, for example, the family relations and stuff like that, which might not appear in the medical or financial applications. So, right, so if you talk to machine learning people, they're of course super excited about providing such services and training these models and everything, and you rarely talk about the privacy aspects. I mean, by doing neural networks, linear mixed models, trees, you can do amazing machine learning models, and you can use these to provide all kinds of predictive services. But then the privacy is a huge concern in many of these. So here's a very simple kind of service that we're imagining, where you would encrypt your genome and you would send it to a cloud service. Maybe the cloud service is Microsoft, maybe it's some other company, and you hope that this cloud service would compute some risk. For example, that you get some kind of disease, or maybe it does some kind of genomic prediction. Or this could be some kind of other medical prediction. It doesn't have to be genomic. But then there is this obvious question, like who else is going to see your DNA sequence during this prediction? And maybe who else is going to see the result? You know, maybe in an extreme case, like there could be a malicious admin at the company who just like steals these records that you sent to them. And if things go like really horribly wrong, you try to apply for your maybe health insurance, and now your genome or your medical information has been leaked, and they say that, okay, well, no health insurance for you, or, well, it doesn't look like you're someone who could do this job, so based on your genomic information, so sorry, no need to come to an interview, or something else. I mean, just, it's still like, I think that at this point, we still don't even understand how much information we can get from the genome. It's like so new still. So it seems like there is this huge duality between data utility and privacy. So yeah, maybe you have tons of data, tons of like really detailed data. Maybe you're the owner of some hospital and you have all these patient records and everything, and you would want to, maybe you would want to train some machine learning models or something. You would want to perform some kinds of computations on these. Or maybe you're an individual, you have your genome sequenced and you would want to get some predictions done on these. So you have data, but you know, you have to like pay a price in privacy to get the utility out of that. And like was mentioned in the previous talk, or previous talks, one of the ways of solving this problem, at least partly, is using encryption. Now, unfortunately, it's not totally trivial at all, because typically, typically when you take some data and you encrypt it, and you try to do some kind of computation on it, well, out comes nonsense, because you can't just take some AES encrypted data and do anything with it. It's completely garbled. So in this sense, like whenever you send your data to a cloud service, they would have to have the ability to first decrypt it. And then they can do whatever computation they do. Maybe they train a machine learning model. Maybe they do a genomic prediction or medical prediction. But there are some very special types of encryption schemes that can do this. So this is sort of a schematic explanation of what we want to do. So we want to use a very special encryption technique, namely homomorphic encryption. We want to encrypt our, or you rather want to encrypt your genome with this homomorphic encryption, send it to a cloud service where it stays encrypted at all times. It's at no point decrypted. The cloud does not have any key information. They can still apply a function to it, a computation, and they get an encrypted result, and they want to send it back to you. So this is, in fact, sort of possible. So I'm going to talk about this and explain some of the problems here and some of the possibilities here. But the technique that allows such a thing is called homomorphic encryption. So this is a very recent idea by, well, originally by Greg Gentry, from 2009 to construct such an encryption scheme that allows you to do these computations. Now, how does that actually work? Well, the idea is that if you own the private data in the upper left corner, you can either compute a function on it and you get f of x, but again, in these scenarios that I explained, you might not know the function. Like say this is some genomic prediction. Like you don't have the genomic prediction model. You haven't spent, you know, $5 million developing this model. So instead, you encrypt your data, and now, by the power of this homomorphic encryption, the cloud service, you send it to the cloud service, and they can compute the function on the encrypted data. And when you decrypt it, you actually get the f of x out from this. So at no point the service provider sees any information. Unfortunately, the restriction, or the restrictions here are rather strong. Namely, we can only compute polynomials Okay, well, not too bad. We can only compute low-degree polynomials, let's say, rather efficiently. But once you have to compute higher-degree polynomials, it gets increasingly difficult. So, well, of course, we want to try this, and we want to study this in practice, and we want to run some examples and stuff like this. So we had to develop, or we decided to develop our own homomorphic encryption library called SEAL, Simple Encrypted Arithmetic Library. So it's actually freely available. So if you want to try it, you can download it from sealcrypto.codeplex.com. It's written in C++, and there's .NET wrappers available if you like C#. And we're sort of actively working on developing this. And so it's like focusing on, I would say, good engineering style, good programming style, and good API design. And it has no external dependencies, so it's very easy to start to use. So here's some example, but I'm actually going to skip that because there isn't that much time. Instead, I want to demonstrate some things that we can do, because that's much more interesting than hearing about the restrictions. So, interesting. Okay, here. So, the first example that we decided to implement using this SEAL library is this genomic prediction using a linear mixed model. So these are models that are used all the time for genomic risk prediction. In this case, we're not predicting any risk. We're predicting the flowering time of a flower from its genome. And in this case, we use 200,000 locations in the genome, something like up to 1 million locations, up to 10 million locations in the human genome is totally reasonable and sort of a practical scale. So this 200,000 is totally practical scale. So what's going on here is that the flower's genome is encrypted using homomorphic encryption. It's stored in a service, in a cloud service. This is what we would imagine doing here. If we had such like a genomic prediction service, the genome of the customers, they would be stored in the cloud service, encrypted using homomorphic encryption, and now the customer requests a query. So let's request a query. How long does it take for this flower to, or this plant to produce a flower? Well, that didn't take too long. We sent the request to the server. The server did 1.5 seconds of computation on these 200,000 locations in the genome, and sent the result back to us, and we decrypted it, and we got 29.1 days expected time. So, you know, if you use 1 million locations, 10 million locations, it's just you just scale the time basically by that, however much you want, going up from 200,000 locations. And this gives like perfectly good accuracy. There is no accuracy loss per se happening when you use homomorphic encryption. If you don't like Genome, or I don't know if that makes sense, but if instead you want to see some medical risk prediction, we have another example where we wanted to predict how dangerous pneumonia is for a patient who has been admitted to a hospital, or who comes to a hospital and is diagnosed with pneumonia. We want to like know how dangerous is that to the patient. And this kind of prediction can be done pretty accurately based on some simple medical attributes. So here we have, I think, 40 different medical variables that the hospital can measure. And we simply apply a degree 4 polynomial model. Again, because we're using homomorphic encryption, we have to use polynomial models because that's all we can evaluate on encrypted data. But turns out that with this model, we get actually really good accuracy, or area under the curve, which is the correct measure of accuracy here. So we get like really good performance with such a model. And again, it took like 8 seconds to do this computation, 1 second to decrypt, and again, by the power of homomorphic encryption, we actually did a little trick here. We batched together 4,000 medical records into one. So there's actually 4,000 patient cases here done simultaneously. So I can just like skip on to the next one. The slider here tells how whether the patient should go home to eat chicken soup or whether they should be really admitted to the hospital. So such predictions, these genomic predictions, these medical predictions are totally reasonable, totally possible to do with homomorphic encryption. And it's just a few seconds. And the question, like, of course, if you do it on plain text data, it's like some milliseconds. But like, the question is not so much about that time difference. The question is, are you willing to wait this like 8 seconds or 2 seconds to get your medical risk prediction or your genomic prediction? Or is that, is that an acceptable time to you? So I believe I'm pretty much done.
Speakers come back. We can take some questions from the audience, or I have some questions as well. I think there's a question over there. Does somebody have mics to run around?
I can just yell loud. So it's kind of an odd question, but you put up that list in the final talk. You put up that list of like, what's the worst case scenario for someone getting their hands on genomic information, right?
Mm-hmm.
But like, isn't that ultimately a question of laws and government? Because we've been able to, if you wanted to, you could discriminate against people based on genomic information for years, like how much melanin you produce, your X chromosome count. I mean, there's a lot of traits at which, clearly the only solution is legal, not, hiding the fact that, you are who you are. And someone who wanted to be a bad actor, your DNA is very easy to get. I can, shake your hand and collect some.
Yeah.
So, what am I missing?
So just really quick, the question was sort of, are there problems here that we should be thinking about from a legal perspective rather than from a technological perspective?
Right. So maybe you're not missing anything. Possibly not, but like, possibly you are. Like, so suppose there are these genomic testing services, for example, and they operate for 15 years. Now, how many patient records do they have there, right? Like, how many patient records go through them? If this kind of database is leaked, that might be kind of problematic. Of course, what you say is completely true. It's really easy to get, if you decide, okay, you are my victim, I'm going to get your DNA. Yeah, that's very easy.
I would say it's a matter of scalability, right, of the attack. So, I mean, it's much cheaper and, more damaging to hack into a database than target single individuals, right? You actually need physical access to them, or at least, let's say, my glass over there, or my hair that I might have shed while listening to the talks, right? But I mean, if I can add one thing, I think I agree. I think in the sense that we do need also legal and policy regulations, which unfortunately we know tend to lag behind very often. But I think we need both. I think we need the kind of technical protection of data, but also the legal ones. And what we don't want is having privacy fears towards progress in genomics, right? So if we can help with that, maybe it's a good thing for everybody.
Right.
Got a mic coming.
Usually I don't have too much trouble projecting my voice, but I'll use the mic anyway.
Recording.
Sure.
Oh, it is recording. Okay, great. So I got 2 questions. One starting with something that Emiliano said in his talk, the nature of not even protecting the data, but also protecting the tests that are being run on the data. I forget the exact wording you had, but I think that was the general context of what you're
Secret
saying.
sauce or something. Well,
I'm sorry?
the word that I used?
Yeah.
Or I think secret sauce or test specifics. Or,
Okay, yeah. So we're in the vernacular either way. So Is that an actual need in the industry? I mean, because my understanding, and I am a computer scientist, not a medical person, that a lot of these tests had to go through some sort of FDA vetting or some sort of public vetting, and that they have to be out there anyway. I mean, other than probably very experimental testing, is that a real perceived need in the community?
Right. So I mean, I would answer with one case that actually happened, is when Myriad Genetics tried to patent a specific test, one for BRCA mutation, which is associated to breast and ovarian cancer. So that's, the test is very simple in that you look at, I think, a dozen possible mutation in a specific gene. Right, so they tried to patent that, and the Supreme Court told them you can't patent this because you're essentially trying to patent a human genome, right? You try to patent the gene, right? So I mean, in that setting, the only other possibility for them is to keep the locations that they go and test confidential. I mean, they have to disclose it to the FDA if they're planning to use it, on real patients, of course. But they might not want to disclose it openly and say, okay, now I can, if I have access to my own data, I just do the test myself, right? The open source or everybody can do it, right? So I mean, it might be an actual requirement in this setting when you have companies or pharmaceutical companies that have to protect their intellectual property, let's say. I mean, I don't know if you agree.
Yeah, I think that's a real need. Because, for example, like a pharmaceutical company want to access some data center, and the information they are going to check is actually related to what they are going to do research on. It's very sensitive, so they may not want the data center to know what they are actually looking at.
I had a short question for Kim and a longer question for Xiaoqing. Carl Gunter from the University of Illinois. Kim, on your thing, I wonder how much has it been studied to find out whether there are any side channels that are problems for these homomorphic encryptions? For example, you might imagine an algorithm that terminates very quickly if it finds a high risk. Maybe there's some dominant traits and it looks for those, and if it finds them, it stops. And so like you'd see different run times for different kinds of results.
Okay, so the algorithm running on the server, on the cloud service, in the cloud service, that has to have the same runtime no matter what, because it has to touch basically all of the data, and it has to do the same computation no matter what the encrypted message is. So in that, there cannot be anything like that. It's because if somehow the runtime depended on the underlying message, then yes, it would be leaking. It would be possibly leaking a huge amount of data. Who knows? But that's not the case. It in fact does the same computation no matter what, same operations no matter what.
Mm-hmm. So in other words, you'd think there's a low risk of a side channel like that. But are there actually mathematical proofs of this, like there are of the other things?
Yeah, there's like a mathematical proof, I would say. I mean, you have to do the same operation at every time. It's like a property of the encryption scheme that the ciphertext don't leak anything about the message. Yeah, but I mean, there could be other types of attacks, and I think it is definitely true, or at least I feel like it's true, that homomorphic encryption has been studied a lot from this kind of very cryptographic point of view, and it has been studied less in the context of such complicated scenarios where you have these protocols.
Yeah, it seemed like given your success, that might be a next step, is to look at some of that if it hasn't been done yet. I had one more question for Jacques. Okay, so I was looking at your contest,
Yeah.
and I was involved in thinking about the beacon problem. Okay,
Yeah.
so maybe for the audience you could repeat what the beacon problem is, but A thought I had was for some of these things, non-technical solutions might be much easier than technical solutions. So for example, with the beacon problem, NIH has a system for people to kind of register themselves as institutions.
Right.
And so if you could simply authenticate to some degree the parties that are using the beacons, then that might be much better than any kind of technical solution you could deploy. And so what I was wondering is there any way for your contest to take into account, maybe there's some no-brainer non-technical solution, and then your guys are like, you know, trying to show you can take it down from 15 hours to 14 hours, and, you know, so is there a way to, like, incorporate non-technical issues in that?
Yeah, actually, this is a very good question. So let me recap on what a beacon is. Beacon is a service provided from G4GH, Global Alliance for Global Health. So they have alliance of like over 100 genomic database all over the world. What the service can provide is you ask at which chromosome, which position, if you have a variation, and then each database answer yes, no, not found. And that's all. But it's given you have so many SNPs or so many variations, you can keep on asking this question, and you build a binomial test, and then essentially you can identify whether the target of interest is within, for example, an autism beacon, and then that leaks sensitive information. So we actually considered there's simple solutions. If we use EI Commons or OpenID to authenticate people, then we know we can trace their pattern. Actually, we can compare this with normal patterns. But we talked to the beacon organizers, and they were thinking they still want to keep that public service available to the public. So we need a way. I think we should have a tiered way to protect this service. So for example, if you want allele frequency, the number of query you need to re-identify individual information is much less than it just answer yes, no, not found. So in that sense, they are actually enforcing people to authenticate with EI Commons. They just don't enforce that on the yes, no answers. But we have some technical solutions, I think, that we can trace how much has been reviewed on individual people and whether we want to keep that person in the database or not. So this year, we actually have some innovation award for solutions that are not forced into the traditional technology realm, and we encourage those to be submitted, and we will consider giving award to those solutions as well. Maybe there's some very easy, no-brainer solutions that solve the problem.
Like using EA Commons.
Yes.
Do we have other questions from the audience?
License for the source code of seal, how that code can be reused for for specialized purpose like biomedical applications. Second question is, what do you think? I mean, my understanding the seal is for general purpose for homomorphic encryption. How do you think that the performance is in terms of the memory usage or speed can be optimized towards certain specific function evaluation? Or what do you plan to do any kind of applications that kind?
Okay, so first of all, about the license. So it's published under this MSR license agreement, so it's freely available. You can use it for academic purposes, I think freely. I mean, the license is there on the website, and I don't exactly know what it says, but it's pretty short, and I think it's just like for academic purposes, it's freely usable.
Will the code be reused or reused in the other application?
Can you or can you like change it and then publish it again? Mm-hmm. Well, probably not. Probably not. Okay. If you would want to do something like that, and there's like a good reason for you to want to do something like that, you should contact us and we can set up some other way. And for the other question, so this library actually hasn't been in development for very long. And we've mostly, until very recently, we've focused on like making it really, really easy to use and focused on like perfecting the API and things like this. Only now we are starting to like focus more on the performance aspect. So all these results, in fact, are done with, that I showed you, are done with an older version of the library. In fact, we haven't rerun them with the newer version, which was just released some weeks ago. So there's huge sort of general performance improvement happening, and it's going to be happening for probably a few more months at least. The results that you saw, the performance results, are the result of tweaking the encryption parameters to be particularly suitable for the tasks that we were doing. So sort of the results that we got are good, or pretty good, because we know kind of well what we're doing. If someone who doesn't really know how to do much homomorphic encryption would start doing it, they would do it maybe in the sort of most obvious way. Maybe it wouldn't be quite that fast. It would take maybe some work to improve it, to like fine-tune the parameters. But it wouldn't be probably that far off. But in any case, typically in homomorphic encryption, you always want to tune the parameters and sort of tune the system for your specific computation. So that's how homomorphic encryption works these days. You can do more, like in theory, you can do more general, something called fully homomorphic encryption, where you actually can do an unlimited number of operations. You can do anything, but that's only in theory. In practice, we always kind of fine-tune it a little bit, and if you want to get these really good results, then you have to fine-tune it maybe a little bit more, and that might require some expertise. But basically, getting sort of acceptable results for many simple tasks, many simple computations, should be very easy with the current API of the library.
Do we have additional questions from the audience?
Do you have any comparison with existing homomorphic encryption library like HLE developed by the IBM team in terms of the performance? My question is, do you have any comparison with the existing homomorphic encryption library like the HLE developed by the IBM team?
Okay, so currently we have not performed such comparisons. So different homomorphic encryption libraries sort of focus on a little bit different things, and we are heavily focused on, or heavily focusing on, encrypting integers at a time. Like maybe some other libraries are better suited for encrypting bitwise things, and they have functionality that is really useful for such things. We are focusing really heavily on encrypting integer by integer. So the different libraries have different pros and cons, and I'm sure at the moment IBM's library is probably way faster. They're using you know, some number theory libraries for arithmetic that has been in development for like, you know, a decade. I don't know. I mean, at this point we haven't done any comparisons yet, but once we find a good benchmark, then maybe. It's actually not that easy to come up with even a good benchmark.
So, Kim, actually, I've got a couple more questions about your library and exciting stuff, that exciting work that you'll be doing. So, you know, you're exactly right. All these various libraries have their all kind of various benefits and trade-offs and parameterization challenges, and they're just kind of a mess to use, and it takes like a team of PhDs to figure them out, which is great. You know, you're a team of one. Good for you. Maybe there's other people behind these curtains also. So what scheme did you implement? And then you said you're optimizing for integers. And I'm also very curious about your application data. So the flower example, I mean, what did you actually encrypt? I mean, that there's a lot of data that's out there. Is it a VCF file? Is it something else? Particularly if you're encoding some of that data as integers and then running computations. And I even saw the pneumonia example that you're outputting a floating point number, which is not an integer also. So there's probably some tricks that you're probably learning about how do you encode data, and then that encoded data, what you're actually running on the computation, and what actually are you running for that computation to get those kinds of outputs that you are.
Okay, so first of all, the encryption scheme that we use is the Van Vercauteren scheme, which is scale invariant homomorphic encryption scheme with security based on the hardness of RLWE. So as far as we know, RWE is a very hard computational problem to solve using these parameters that we have, that we're using, and at the moment we have reason to think that it's secure. So that's what we're using, the Funware Cultural scheme. There are other schemes like the BGV scheme used by IBM's library, for example, and the performance of these is probably pretty much the same. They're very, very similar. They just do things in a slightly different order, a little bit different. For the other question about what exactly was the data like, well, okay, now I have to say that I wasn't the genomics person who was working on the flower thing, so I can't remember what the data looked like, but presumably it was just integers 0, 1, 2 for these SNPs, these locations in mutations. And then we had some rational number coefficients in this model. Now, what we did here was we in fact scaled the rational numbers to be integers. Now, we also have some methods of directly encrypting rational numbers in SEAL. This technique has some downsides, and actually we're currently actively working on some solutions to those downsides. So there is a way of, there are several ways proposed in actually pretty recent research papers of encrypting rational numbers directly. SEAL uses one trick of doing that. It's currently maybe not as good as it could be, but we're actively working on a solution that we have. But for the moment, we've seen that typically it's better in many of these, like, especially when you want like really high performance results, you almost always want to probably scale to integers. But we'll see where this goes, because we have some interesting ideas about this.
Are there other questions from the audience? Okay. So I have one question for all of you, I guess, maybe a sort of a high-level question. What do you think is the most sort of promising area for some of these privacy techniques to be deployed? Like what application scenarios do you think will be the first place that will be deploying any of these different techniques?
So I can start. I think like clinical genetic testing is one of the most interesting scenario. So for example, kids with rare disease can deposit their genome in the cloud, and then when the new biomarkers gets revealed, they can be tested in encrypted manner, and then we can constantly feedback to the parents if there are new findings relate to their kids. So that could be a good scenario.
I mean, if I might add another one, is I think we need to follow closely this kind of direct-to-consumer genomics area. Now that 23andMe or many others have sequenced millions of people in just a few countries where they operate, and also, for instance, 23andMe allows you to actually get the raw data that they produce. And so now you can even share it with researchers using, the Apple Health Kit. So I think, this data is going to be out there. So there is opportunities for us as researchers to look for ways to protect it, but also, for researchers and also doctors, right, to tap on this data and, mix it with your medical information, as environmental factors. To kind of, realize this dream of personalized medicine. So probably it's not something immediate, but my personal prediction would be that, 5 years from now, this is going to be a reality, if not less. So. And
you think privacy will be a component of that?
Well, I hope so. Yes.
Yeah, I really think that if we could get inference and machine learning, like the learning component of machine learning, to work very efficiently on encrypted data, or at least on like shared sort of private data across several hospitals. If we could get that to work, if we could train things like trees or random forests, or if we could train neural networks, that would be incredible. Like we could do so much. There could be so many, because you could have so much data coming from like hospitals all around the world. So that would be really, really incredible.
Thanks. So unless there are any other questions, great. I think maybe we can thank all three of our panelists.
Thank you.
Thank you all for coming.